What if the echoes of ancient sea voyages, kinship, and conflict were not just stories, but a living code embedded within your own DNA? Could the genetic markers of Norse raiders, Gaelic mothers, and cosmopolitan crews reveal the true, complex story of who the Vikings were? How did their journeys of conquest and settlement permanently resculpt the genetic map of Europe, leaving a legacy that science is only now beginning to fully decipher? To comprehend the genetic legacy of the Vikings, one must first delve into the deep ancestral tapestry from which they emerged, a complex weave of migrations, adaptations, and regional isolations that defined the Scandinavian peninsula long before the first longship set sail. The genetic foundation of the people who would become known as Vikings was not forged in a vacuum, but was the culmination of thousands of years of demographic history, beginning with the post-glacial recolonization of northern Europe. The earliest inhabitants were Mesolithic hunter-gatherers, genetically distinct populations who followed the retreating ice sheets northward around 11,000 years ago. Studies of ancient DNA, ADNA, from this period reveal a populace with a dual ancestry, drawing from both Western hunter-gatherer, WHG, groups moving up from the south and Eastern hunter-gatherer, EHG, groups migrating from the northeast, across the vast Eurasian plain. These two streams met and mingled in Scandinavia, creating a unique Mesolithic gene pool adapted to a harsh, cold environment characterized by coastlines rich in marine resources and dense inland forests. Their genetic signatures, particularly mitochondrial haplogroups like U4 and U5, form the deepest foundational layer of the region's ancestry. The pivotal shift in this landscape occurred with the arrival of Neolithic farmers around 4000 BCE, who brought with them not only agriculture and animal husbandry, but also a new stream of ancestry originating from Anatolia. This wave of migration, associated with the funnel beaker culture, introduced a genetic profile that had been slowly spreading across Europe for millennia. This influx did not entirely replace the indigenous hunter-gatherers. Instead, a long and complex period of admixture ensued, lasting for over 1,000 years. For centuries, these two distinct groups coexisted, sometimes clashing over resources, but ultimately integrating, resulting in a new, blended gene pool that carried the legacy of both traditions. This fusion is critical to understanding the later Viking population, which was neither purely one nor the other but a product of this ancient synthesis, combining the robust adaptations of the northern hunters with the agricultural technologies of the southern farmers. The subsequent late Neolithic and early Bronze Age, beginning around 2800 BCE, saw the arrival of a third significant ancestral component associated with the corded ware culture, which was in turn linked to the Yamnaya people of the Pontic-Caspian steppe. These semi-nomadic pastoralists introduced Indo-European languages, new burial customs, and a distinct genetic profile, most notably the Y-chromosomal haplogroup R1a and R1b. This migration wave further homogenized the Scandinavian gene pool with that of Central and Eastern Europe, yet the region's relative geographic isolation, bounded by the Baltic Sea and the North Atlantic, allowed for the development of a distinct northern genetic profile through processes of genetic drift and local selection. By the dawn of the Iron Age around 500 BCE, the population of Scandinavia had stabilized into a relatively homogeneous, yet regionally distinct, genetic landscape. The three primary ancestral streams, Mesolithic hunter-gatherers, Neolithic farmers, and Bronze Age pastoralists, had coalesced into the Germanic-speaking peoples described by Roman authors like Tacitus. Within this broader Scandinavian identity, subtle but significant regional differences began to emerge, driven by geography and trade patterns. The peoples of what would become Denmark and southern Sweden were more genetically connected to continental Europe, reflecting their proximity and overland trade routes. In contrast, the inhabitants of Norway's fjord-riven coastline maintained stronger connections across the North Sea, while the populations of eastern Sweden and the Baltic Islands looked eastward, engaging with the communities of Finland and the Eastern Baltic. It was from this complex, layered, and regionally structured gene pool, a mosaic of ancient hunter-gatherers, Anatolian farmers, and steppe pastoralists, forged by millennia of isolation, adaptation, and interaction, that the individuals who initiated the Viking Age would ultimately emerge, carrying within their DNA the entire preceding history of Northern Europe as they embarked on their world-changing voyages. The dawn of the Viking Age in the late 8th century heralded a dramatic expansion of Scandinavian peoples, and the genetic trail left by the first raiders provides a vivid, high-resolution account of their origins, movements, and social structures. Far from being a monolithic horde, these early Viking warbands were complex, often multi-ethnic enterprises, a fact that traditional historical and archaeological sources could only hint at, but which ancient DNA (ADNA) analysis has now unequivocally confirmed. The initial wave of attacks, targeting coastal monasteries and settlements in the British Isles, was predominantly launched from Western Scandinavia, particularly Norway. Genetic studies of both ancient remains and modern populations in areas of primary Viking impact, such as the Northern and Western Isles of Scotland, the Isle of Man, and coastal Ireland, reveal a strong prevalence of Y-chromosomal haplogroups like R1az284, a lineage with a distinct Norwegian signature. This genetic evidence corroborates the historical narrative of the sagas, which depict Norwegian chieftains leading expeditions to plunder and later settle these lands. A landmark 2020 study sequencing the genomes of 442 individuals from Viking Age sites across Europe provided unprecedented detail. It revealed that the warband was a fundamental unit of Viking society, and that these groups were often composed of close kin. For example, a mass grave of executed Vikings at Ridgeway Hill in Dorset, England, dating to the early 11th century, was found to contain a group of young men primarily of Danish and Danish-like ancestry, suggesting a cohesive war party from a specific region. Similarly, analysis of remains from a famous boat burial in Somme, Estonia, dated to the mid-8th century, predating the canonical start of the Viking Age, showed a group of elite warriors from central Sweden who were closely related, including 4 brothers buried side by side. This genetic evidence for kinship-based raiding parties revolutionizes our understanding of their organization, suggesting that loyalty and family ties were the glue that held these formidable military units together. Furthermore, the genetic data has shattered the myth of a pure Scandinavian identity for the Vikings. While the leadership and core of these groups were indeed Scandinavian, they readily incorporated individuals from other regions. Skeletons buried in Orkney with full Viking grave goods were shown to have genetic profiles indistinguishable from contemporary Pictish and Irish populations, indicating that local individuals could be adopted into Viking society and achieve high status. This genetic mixing was not a later development but was present from the very beginning. The Viking phenomenon was as much a cultural and economic one as it was ethnic. To go Viking was a career choice open to ambitious individuals from various backgrounds who were drawn to the wealth and prestige offered by raiding. The genetic trail also differentiates the major streams of Viking expansion. While Norwegians dominated the North Atlantic route, the large-scale armies that began to campaign in and colonize eastern England from the mid-9th century were primarily of Danish origin. Their genetic footprint is more subtle, as the Danes were genetically closer to the Anglo-Saxons they encountered, both being part of the broader Germanic family of peoples. However, sophisticated statistical methods analyzing shared segments of DNA have identified a clear, albeit smaller, contribution to the English gene pool in the regions of the former Danelaw. The very first raiders were the vanguard of a movement that would permanently alter the genetic map of Europe, and their remains tell a story not just of violence and conquest, but of kinship, multiculturalism, and the complex social dynamics that propelled small Scandinavian communities onto the world stage. The establishment of the Danelaw in the latter half of the 9th century represents a pivotal moment in the history of England, marking a transition from coastal raiding to large-scale settlement and colonization by Scandinavian peoples, primarily of Danish origin. This demographic shift, where the Great Heathen Army evolved from a mobile fighting force into a settled population of farmers, traders, and administrators, left an indelible and measurable mark on the genetic landscape of northern and eastern England. For centuries, the extent of this settlement was debated by historians, with some arguing for a small-scale elite takeover and others for a mass migration of Scandinavian families. Modern genetic studies have provided a decisive, albeit nuanced, answer, revealing a significant and lasting Scandinavian contribution to the English gene pool that points towards a substantial folk movement. The People of the British Isles Project, a comprehensive genetic survey, was able to identify distinct clusters of genetic variation within the modern British population. One of the most striking findings was the identification of a genetic stratum in Yorkshire and surrounding areas that showed a clear affinity with modern Danish and northern German populations, a signature distinct from the primary Anglo-Saxon genetic layer found elsewhere in England. This evidence strongly suggests that the Viking settlement was not merely a military occupation but involved a considerable number of Scandinavian settlers who integrated with the local Anglo-Saxon population, forming a new, hybrid Anglo-Norse society. The process of integration is key to understanding the genetic legacy. Rather than a wholesale replacement of the existing population, the evidence points to extensive intermarriage. The Vikings did not arrive in an empty land. They settled alongside and among a numerous Anglo-Saxon peasantry. Over generations, the two groups, who were already culturally and linguistically similar, merged. This fusion is reflected in the language, with the adoption of countless Norse words into Old English, and in the legal and social structures of the region. Genetically, it created a population that was a mosaic of the two ancestral groups. While the overall Scandinavian ancestry in modern English people is estimated to be around 6%, in the core areas of the Danelaw, such as Yorkshire and Lincolnshire, this proportion is significantly higher, likely reaching 10 to 20% in some communities. This figure, while not representing a majority, is far too large to be explained by a small warrior elite and implies the arrival of thousands of Scandinavian men, women, and children. The analysis of Y-chromosomal and mitochondrial DNA adds further detail to this picture. The paternal lineages show a clear influx of Scandinavian haplogroups like I1 and specific subclades of R1a, which became established in the Danelaw. The maternal lines also show a Scandinavian contribution, indicating that entire families made the journey across the North Sea. This contradicts the simpler model, seen in some other Viking colonies, of Scandinavian men exclusively taking local wives. The Danelaw was a true colonization effort. The genetic data aligns remarkably well with other forms of evidence, such as the dense distribution of place names with Norse origins, those ending in suffixes like by village, thorp outlying farmstead, and thwaite clearing. A map of these place names is a virtual map of Scandinavian genetic influence, demonstrating a deep and lasting demographic impact on the countryside. The creation of the Danelaw was therefore not just a political event but a profound biological one, a large-scale admixture event that permanently enriched the genetic tapestry of England and forged a new regional identity that was neither purely Anglo-Saxon nor purely Scandinavian, but a resilient and dynamic fusion of the two. The Viking impact on Ireland and the lands bordering the Irish Sea engendered a unique process of cultural and genetic fusion, creating a vibrant and powerful hybrid society known as the Norse-Gaels or Hiberno-Norse. Unlike the large-scale agricultural settlement of the Danelaw, the Viking presence in Ireland, initiated primarily by Norwegian adventurers, was initially focused on establishing fortified coastal bases, long forts, such as Dublin, Waterford, Wexford, Cork, and Limerick. These sites, which evolved into Ireland's first major towns, served as centers for raiding, and more importantly, for a vast and lucrative international trade network that included the trafficking of slaves. This urban, trade-focused model of settlement fostered a different kind of interaction with the indigenous Gaelic Irish population, leading to a distinct genetic outcome. From the outset, the interaction was characterized by both conflict and alliance, but crucially by extensive intermarriage. The Viking settlers were predominantly male, and they frequently formed alliances with Irish kings by marrying into Gaelic noble families. Conversely, many Gaelic women, both high-born and commoners, became the wives and concubines of Norse settlers, a process that is starkly visible in the genetic record. Studies of the Y-Chromosome, paternal line, in the modern Irish population reveal a significant, though regionally variable, Scandinavian contribution, with specific haplogroups of Norwegian origin being more common in the areas around the Old Norse city-states. However, the maternal story, told through mitochondrial DNA, mtDNA, is overwhelmingly one of Gaelic continuity. This pronounced parental asymmetry, Norse fathers and Gaelic mothers, is the genetic hallmark of the Norse-Gaelic synthesis. This process gave rise to a new warrior aristocracy that was bilingual, bicultural, and immensely influential. Powerful dynasties with mixed ancestry, like the Y-Chromosome, the House of Ivar, came to dominate not only the Irish Sea but also ruled territories in Scotland, the Isle of Man, and even Northumbria. These rulers and their followers did not see themselves as exclusively Norse or Gaelic. They were a new entity, embodying a fusion of the two cultures. This is reflected in the material culture found in archaeological digs in Dublin, which shows a blend of Scandinavian and Irish artistic styles, and in personal names recorded in the Irish annals, where individuals frequently bore both a Norse and a Gaelic name. The Isle of Man stands as a prime example of this synthesis. Genetically, the modern Manx population shows one of the highest concentrations of Norwegian-linked Y-chromosomes outside of Scandinavia, yet its foundational maternal lineages are predominantly from Ireland and Scotland. The island's political and linguistic history mirrors this duality. The slave trade, operated by the Vikings out of Dublin, also played a significant role in shaping the region's genetics. These slave markets trafficked people from across the British Isles and even continental Europe, introducing a diverse array of genetic lineages into Ireland. While many were sold on to other parts of the Viking world, such as Iceland, many remained, further diversifying the gene pool of the Norse-controlled towns. Over time, the Norse settlers in Ireland became increasingly Gaelicized. They adopted the Irish language, converted to Christianity, and became integrated into the complex web of local politics. By the time of the Anglo-Norman invasion in the 12th century, the descendants of the original Viking settlers were often indistinguishable from their Gaelic neighbors, yet their genetic legacy endured. The Norse-Gaelic synthesis was a dynamic process of admixture that created a new, powerful cultural and political force in the Irish Sea world, leaving a permanent genetic imprint that testifies to a history of interaction that was far more complex than simple raiding and pillaging. While the most intensive Viking genetic legacies were forged in the British Isles and the North Atlantic, two remarkable southern expansions, one west into France and the other southeast to the heart of the Byzantine Empire, created distinct and far-flung echoes of Scandinavian DNA. The first of these ventures resulted in the formation of Normandy. In the early 10th century, the West Frankish king, Charles the Simple, granted a sizable territory at the mouth of the Seine to a Viking leader named Rollo and his followers. This strategic concession aimed to put an end to their incessant raiding by integrating them into the Frankish political system. The Northmen who settled this land, which came to be known as Normandy, were primarily of Danish and Norwegian origin. However, their demographic impact and subsequent genetic legacy were fundamentally different from that seen in the Danelaw. The number of Scandinavian settlers in Normandy was likely much smaller, constituting an elite warrior class that ruled over a numerous native Gallo-Roman and Frankish population. Consequently, the process of cultural and linguistic assimilation was astonishingly rapid. Within a few generations, the Norse settlers had abandoned their Old Norse language in favor of the local Romance dialect, which would evolve into Norman French, and they adopted French customs, laws, and military techniques, most notably cavalry warfare. This cultural assimilation was so complete that by the time of the Norman conquest of England in 1066, the invaders were French-speaking Christians. Genetically, this meant that the Scandinavian DNA of the original settlers was quickly diluted within the much larger local gene pool. While a subtle genetic layer of Scandinavian ancestry can still be detected in the modern population of Normandy, particularly in paternal lineages, it is not as pronounced as in the Danelaw. The true significance of the Norman genetic legacy lies in its second chapter, the conquest of England. When William the Conqueror's army crossed the Channel, it was, in effect, reintroducing a strain of Scandinavian DNA back into the British Isles. The Norman aristocracy that replaced the Anglo-Saxon elite was therefore a complex mix of Frankish, Gallo-Roman, and diluted Viking ancestry. This event added another intricate layer to the genetic makeup of the English people, a final wave of continental DNA that carried with it the ghost of its Viking origins. The second southern echo traces the path of the Varangians, Scandinavian warriors who traveled the great river routes of Eastern Europe to seek their fortunes in the wealthy and sophisticated Byzantine Empire. Many served as elite mercenaries in the personal bodyguard of the Byzantine emperor, the legendary Varangian Guard. These men, mostly from Sweden but also from Norway and Denmark, were renowned for their loyalty, strength, and ferocity in battle. While many hoped to return to Scandinavia laden with riches, a significant number spent their entire careers in Byzantine service, and some settled permanently in Constantinople or other parts of the empire. They married local Greek, Slavic, and Anatolian women, and their children were absorbed into the cosmopolitan society of the Eastern Roman Empire. The genetic footprint of the Varangians is, by its nature, far more diffuse and difficult to trace than that of the Normans. They were a transient military group, not a colonizing force, and their numbers were never large enough to make a significant demographic impact on the vast populations of the Balkans and Anatolia. However, their presence is recorded in Byzantine chronicles, in runestones erected in Sweden commemorating those who died in the East in the Greek lands, and potentially in rare paternal lineages found today in Greece and Turkey that hint at a Scandinavian origin. This legacy represents the farthest southern extent of direct Viking gene flow, a testament to their incredible mobility and the allure of the legendary city they called Miklagard. Together, the Normans and Varangians demonstrate the diverse strategies and outcomes of Viking expansion, leaving behind faint but fascinating genetic echoes in the southern reaches of Europe. The eastern expansion of the Vikings, undertaken primarily by people from what is now Sweden, forged a path not of coastal raiding but of ambitious long-distance trade and political state-building along the vast river systems of Eastern Europe. These Scandinavian merchant warriors, known as Varangians by the Byzantines and as the Rus by the local Slavic and Finnic tribes, navigated the Dnieper and Volga rivers to connect the Baltic Sea with the Black Sea and the Caspian Sea, establishing a lucrative trade route that exchanged northern goods like furs, amber, and slaves for Byzantine silk and Arab silver. This eastern venture culminated in the establishment of the first East Slavic state, Kievan Rus, a pivotal moment in the history of Russia, Ukraine, and Belarus. The genetic legacy of the Rus is a topic of intense study and historical debate, centered on the Normanist theory, which posits a foundational role for a Scandinavian elite in the creation of this state. Genetic evidence has provided powerful, though complex, insights into this question. Unlike the dense family-based settlements of the Danelaw, the Scandinavian presence in the east appears to have been a classic case of elite dominance. The number of Rus settlers was relatively small compared to the vast sea of Slavic and Finnic peoples they lived among and eventually ruled. Their settlements, such as Staraya Ladiga and Novgorod, began as trading posts or gorids, where a Scandinavian military and merchant elite oversaw commerce. The primary genetic impact, therefore, was concentrated within the emerging ruling class. The most compelling piece of evidence comes from the study of the Rurikid dynasty, the royal family that claimed descent from the semi-legendary Viking chieftain Rurik and ruled Kievan Rus and its successor states for over 700 years. Genetic testing of modern-day descendants of this dynasty has identified a specific Y-chromosomal haplogroup, N1C1, also known as NL550, as the lineage of Rurik's paternal line. This haplogroup is most common today in Finland and the eastern Baltic region, but is also found at lower frequencies in Sweden, particularly in the Roslagen area, the historical heartland of the Swedish Vikings who went east. This discovery provides strong support for the Scandinavian origin of the founding dynasty of the Rus state. However, the broader genetic impact on the general population of Russia, Ukraine, and Belarus was far more limited. While some Scandinavian paternal and maternal lineages were introduced into the Slavic gene pool, they were largely absorbed by the much larger native population. The Rus men frequently took local Slavic wives, a practice that accelerated their cultural and linguistic assimilation. Within a few generations, the descendants of the original Vikings were speaking Old East Slavic, had adopted Slavic names, and by the end of the 10th century, had embraced Orthodox Christianity. The genetic signature of the Rus is therefore one of a small but influential stream of Scandinavian DNA that was injected primarily at the elite level of society, helping to catalyze political centralization but without causing a major demographic shift in the wider population. Archaeological evidence from Rus burial sites supports this picture, showing graves with characteristic Scandinavian boat burials and grave goods alongside individuals who were genetically local, sometimes buried in the same cemetery. This indicates a society where a Scandinavian cultural identity was maintained for a time, particularly among the elite, but where integration and admixture with the local population was the dominant long-term trend. The legacy of the Rus is thus a prime example of how a numerically small but well-organized and ambitious group can have a profound political and cultural impact that far outweighs its genetic footprint. The settlement of Iceland, beginning in the late 9th century, stands as one of the most remarkable events of the Viking Age and has created a unique natural laboratory for the study of human genetics. Unlike other areas of Viking expansion, Iceland was a large, uninhabited island, meaning its entire human gene pool was established by a known founding population within a relatively short and well-documented period. This unique circumstance, combined with centuries of relative isolation, meticulous genealogical records dating back to the settlement era, and a modern homogeneous population, has allowed scientists to reconstruct the genetic origins of a nation with unparalleled precision. The story begins around 874 CE, when Norse chieftains, primarily from western Norway, began to settle the island, seeking new land and fleeing the centralizing power of King Harald Fairhair. Historical sources, most notably the medieval Landnamabok, the Book of Settlements, provide a detailed, albeit semi-legendary, account of the first settlers. For decades, this was the only window into the island's origins, but modern genetics has added a rich and sometimes surprising layer of biological data. Comprehensive studies of the entire Icelandic population have revealed a striking and consistent pattern of ancestral asymmetry. Analysis of the Y-chromosome, which traces paternal ancestry, shows that approximately 80% of Icelandic male lineages are of Norse/Scandinavian origin. This aligns well with the historical narrative of Norwegian chieftains leading the settlement. However, the analysis of mitochondrial DNA, mtDNA, which traces maternal ancestry, tells a very different story. An astonishing 62% of Icelandic maternal lineages are of Gaelic origin, from Ireland and Scotland. This stark contrast between the paternal and maternal gene pools paints a vivid picture of the social dynamics of the settlement. The founding population was not a simple transfer of Norwegian society, but a complex mix of high-status Norse men and a larger contingent of Gaelic women. While some of these women may have been the wives of Norsemen who had lived in the British Isles before migrating to Iceland, the sagas and the genetic data strongly suggest that many were brought as slaves or concubines, captured during Viking raids in the Irish Sea region. Iceland was therefore founded by a hybrid population from its very inception. This founding event had profound and lasting genetic consequences. The relatively small number of initial settlers, estimated to be between 10,000 and 20,000 people, meant that the island's gene pool was subject to a strong founder effect. The specific set of genetic variants present in these first arrivals, both common and rare, became the basis for the entire future population. Furthermore, the island's subsequent history of geographic isolation, punctuated by population bottlenecks caused by volcanic eruptions, famines, and plagues like the Black Death, further shaped its genetic landscape through the process of genetic drift, where gene frequencies can change dramatically by random chance. This unique genetic history has made Iceland a goldmine for medical genetics. The combination of a limited diversity gene pool, extensive genealogical records, and a national healthcare system has enabled researchers, most famously the company Decode Genetics, to identify specific genes associated with a wide range of common and rare diseases, from schizophrenia to heart disease and cancer. In essence, the genetic legacy of the Viking Age settlement is still being written in the DNA of modern Icelanders, providing critical insights into human health and disease that are only possible because of this extraordinary North Atlantic genetic experiment. The Norse colonization of Greenland, initiated by Eric the Red around 985 CE, represents the westernmost extent of the Viking expansion and one of its most enigmatic chapters. For nearly 500 years, a thriving European society of farmers and hunters existed on the edge of the Arctic, maintaining trade and cultural ties with Iceland and Norway across a perilous stretch of ocean. The ultimate disappearance of this community in the 15th century has long been a source of fascination and debate, and genetics is now providing crucial clues about who these Greenlanders were, how they lived, and what might have become of them. Ancient DNA, ADNA, extracted from human and animal remains excavated from Norse farmsteads in the two main settlement areas, the larger eastern settlement and the smaller western settlement, has confirmed their overwhelming European origin. Genetic profiles of the Norse Greenlanders are closely related to those of medieval Icelanders and Scandinavians, affirming the historical accounts of their origins. These studies have also offered intimate details about their society. For instance, analysis of kinship has shown that neighboring farms were often occupied by extended families, and the society appears to have been highly inbred, a consequence of being a small, isolated population. This genetic isolation likely posed significant long-term challenges to the health and viability of the colony. One of the most compelling questions is the extent of interaction and interbreeding between the Norse settlers and the indigenous peoples they encountered. The first people the Norse met were likely the Dorset culture, who later disappeared, followed by the Thule people, the direct ancestors of the modern Inuit. The Icelandic sagas, such as the Saga of the Greenlanders and Eric the Red Saga, refer to these people as Skrælingr and describe encounters that ranged from trade to violent conflict. For decades, researchers have searched for genetic evidence of admixture. Extensive genetic surveys of modern Inuit populations in Greenland and Canada have been conducted, looking for telltale signs of European ancestry dating back to the Viking Age. To date, No definitive evidence of Norse-Inuit interbreeding has been found in the modern Inuit gene pool. This suggests that if any genetic exchange did occur, it was extremely limited, and the resulting lineages either died out or are so rare they have not yet been detected. This genetic isolation stands in stark contrast to the extensive admixture seen in Iceland and the British Isles, pointing to a very different and likely more hostile relationship between the settlers and the native population in Greenland. The story of the Norse in the Americas does not end in Greenland. The sagas describe voyages even farther west to a land they called Vinland, now known to be North America. The discovery of the Norse settlement at Lonseux Meadows in Newfoundland provided archaeological proof of their presence. For a long time, there was no genetic evidence of this contact. However, a groundbreaking discovery turned this on its head. Genetic analysis in Iceland identified a specific mitochondrial DNA lineage, C1e, of clear Native American origin within a few Icelandic families. By tracing genealogies, researchers were able to determine that this lineage was present in Iceland as far back as the early 18th century and, based on its unique mutations, was almost certainly introduced to the island around the year 1000 CE. The most plausible explanation is that a Native American woman was taken from Vinland to Iceland by a Norse expedition, and her maternal line has survived there for over a millennium. A single living genetic link to the Vikings' brief sojourn in the New World. This solitary lineage underscores the tantalizing and often mysterious nature of the farthest reaches of the Viking genetic legacy. The popular image of the Viking Age is overwhelmingly masculine, dominated by bearded warriors, male chieftains, and longships crewed by men. However, this perception obscures the fundamental role that women played as migrants, settlers, culture bearers, and the biological anchors of new societies. The story of these women, often absent from the male-centric historical chronicles, is vividly told by mitochondrial DNA, mtDNA, a segment of the genome passed down exclusively from mother to child. By tracing these maternal lineages, geneticists can reconstruct female migration patterns across the Viking world, providing a crucial counterbalance to the paternal story told by the Y-Chromosome and revealing a narrative of complexity, agency, and adaptation. The most dramatic illustration of the importance of female mobility comes from the settlement of the North Atlantic islands. In Iceland, as previously noted, the founding population was a stark mix of Norse men and Gaelic women, with over 60% of maternal lineages originating in the British Isles. This pattern is mirrored in the Faroe Islands, where mtDNA studies show approximately 85% of maternal lines are of Gaelic origin, while the paternal lines are about 87% Scandinavian. This consistent pattern of Norse men pairing with Gaelic women across the North Atlantic points to a systematic and widespread social dynamic. It reflects a world where Viking men, operating from bases in Scotland and Ireland, formed unions, coercive or consensual, with local women and then moved onward to colonize new lands, bringing these women and their children with them. These Gaelic women were not passive passengers in history. They were the founding mothers of these new island nations, and their genetic legacy is dominant to this day. However, the story is not solely one of local women being incorporated into Viking society. There is clear and compelling evidence for the large-scale migration of Scandinavian women as well. In the Northern Isles of Scotland, Orkney and Shetland, which became thoroughly Norse colonies, the genetic contribution is more balanced. Studies show a significant proportion of both paternal and maternal lineages, around 40 to 50% for mtDNA in some estimates, are of Scandinavian origin. This indicates that the settlement of these islands was not just a military venture but a full-scale folk migration involving entire families from Norway. Similarly, in the Danelaw region of England, while intermarriage with local Anglo-Saxon women was common, The presence of Scandinavian mtDNA haplogroups suggests that Danish women also crossed the North Sea to settle and farm the new lands. Ancient DNA from Viking Age burials further enriches this picture, challenging simplistic notions of Viking gender roles. The analysis of high-status female graves, such as the magnificent Oseberg ship burial in Norway, which contained the remains of 2 women, confirms their elite status. More controversially, the re-examination of a famous warrior grave at Burka in Sweden, long assumed to be male due to its full complement of weapons, revealed through DNA analysis that the skeleton was, in fact, female. While the interpretation of this female warrior is still debated, it opens up the possibility that women's roles in Viking society were more varied than previously thought. The mtDNA saga reveals that the Viking expansion was as much a story of women's journeys as it was of men's. Whether they were Gaelic slaves taken to Iceland, Norse farm wives settling in Orkney, or Swedish traders' partners in the East, women were the essential thread that wove new societies together. Their genetic lineages trace a map of interaction, integration, and the creation of new hybrid identities across Europe, demonstrating that without the mobility and resilience of its women, the Viking Age would have been impossible. The Y-chromosome, passed directly from father to son with only minor mutations over generations, serves as a powerful genetic tracer for the male-driven movements that characterized much of the Viking Age. It allows researchers to follow the paternal lineages of Scandinavian men as they radiated out from their homeland, charting their paths of raiding, trading, conquest, and settlement across a vast geographical expanse. The distribution of specific Y-chromosomal haplogroups and their subclades across modern Europe acts as a living map of this medieval diaspora, providing clear biological evidence for the historical accounts of Viking activity. 3 major haplogroups are most strongly associated with Scandinavian ancestry: I1, R1a, and R1b. While these are found across Europe, specific subclades within them are highly concentrated in Scandinavia and are considered robust markers of Viking-era ancestry. The haplogroup I1 is perhaps the most archetypal Viking lineage. It is believed to have originated in Northern Europe and is found at its highest frequencies today in Sweden, Denmark, Norway, and Finland. It spread into other parts of Europe, particularly the British Isles and Normandy, closely mirrors the known paths of Viking migration. When a man in Yorkshire, England, discovers he belongs to haplogroup I1, it provides strong evidence that his direct paternal ancestor was likely a Danish Viking who settled in the Danelaw over 1,000 years ago. Haplogroup R1a tells a more regionally specific story. One major branch, R1az284, is overwhelmingly found in Norway and in the areas of Norwegian settlement. Its presence is particularly strong in the northern and western Isles of Scotland, the Isle of Man, and Iceland. This lineage effectively traces the Norwegian maritime expansion into the North Atlantic. In contrast, other branches of R1a are more associated with the eastern Swedish route. The paternal lines of the Russ elite, for example, connect to different R1a subclades, as well as the distinct N1C1 haplogroup, highlighting the different origins and paths of the various Scandinavian groups. R1b is the most common haplogroup in Western Europe, but again specific Nordic branches like R1b-U106 are part of the Germanic genetic heritage that includes the Vikings. Distinguishing the Viking contribution of this lineage from the earlier Anglo-Saxon or Frankish waves can be challenging, but advanced analysis of shared genetic markers allows researchers to identify the signature of the later Scandinavian arrivals. The genetic data reveals the social dynamics of the Viking expansion. The establishment of these paternal lines in new territories was often a result of elite dominance. A relatively small number of successful Viking warriors or chieftains could have a disproportionately large genetic impact by fathering numerous children with local women. This is evident in the stark parental asymmetry seen in places like Iceland and the Faroe Islands, where a limited number of Norse paternal lines are now widespread, while the maternal diversity is much greater and more Gaelic. The Y-Chromosome trail also confirms the multicultural nature of Viking warbands. Analysis of ancient DNA from Viking cemeteries sometimes reveals individuals with paternal lineages from southern Europe or the Baltic buried with full Scandinavian honors, showing that to be a Viking was a profession as much as an ethnicity. The modern fascination with genetic genealogy has brought this ancient history into the personal realm. Commercial DNA testing allows individuals to discover their own paternal haplogroup, connecting them to this sweeping historical narrative. While carrying a Viking marker does not mean one is a direct descendant of a famous figure like Ragnar Lothbrok, it does signify that one's paternal line was part of the great demographic and cultural upheaval that defined the Viking Age, a direct, unbroken genetic link to the men who sailed the longships and reshaped the map of Europe. The romanticized image of a Viking warband, a homogeneous crew of blonde, blue-eyed Scandinavians, has been thoroughly dismantled by the revolutionary insights of ancient DNA, ADNA. The genetic record reveals a far more complex and cosmopolitan reality, showing that the longships that terrorized and transformed Europe were often crewed by multicultural and multi-ethnic assemblages of individuals drawn from across the Viking world and beyond. The very act of going Viking, Farai Viking, was less an ethnic imperative and more a career choice, an enterprise that promised wealth, status, and adventure, attracting ambitious, opportunistic, and often desperate individuals from a wide array of backgrounds. This genetic diversity was not a late-stage development but a feature of the Viking Age from its very beginning. The analysis of the Somme ship burials in Estonia, dating to the mid-8th century, revealed a war party of elite Swedish warriors, but even within this related group, their isotopic signatures suggested diverse geographic origins within Scandinavia. As the Viking Age progressed, this diversity became even more pronounced. A landmark 2020 study that sequenced hundreds of Viking Age genomes found individuals buried in Scandinavia with genetic profiles matching those from southern and eastern Europe, indicating a constant flow of people into the Scandinavian homelands. Conversely, Viking graves in other parts of Europe revealed a surprising degree of non-Scandinavian ancestry. Skeletons buried in Orkney with full Viking honors, including swords and shields, were shown to be genetically indistinguishable from the local Pictish or Irish populations. This suggests that local men could be fully integrated into Viking warbands, adopting their culture, and achieving high status within their ranks. The Viking crew was a meritocracy of sorts, where prowess in combat and loyalty to one's chieftain mattered more than ethnic origin. This genetic melting pot is starkly illustrated in the analysis of mass graves. The Ridgeway Hill burial pit in Dorset, England, contained the remains of over 50 decapitated Vikings executed by Anglo-Saxons. While their genetic profile was predominantly Danish-like, it also included individuals with ancestry from further east in the Baltic, and even hints of Slavic admixture, consistent with a war party drawn from the fluid and diverse populations of the southern Baltic coast. The great Viking armies that operated in Francia and England were not national armies in the modern sense but confederations of warbands, each with its own leader, and their ranks would swell with recruits from the lands they campaigned in. This cosmopolitanism was a key to their success. It provided a larger recruitment pool and a diverse set of skills and local knowledge. A crew might include a Norwegian navigator, a Swedish runemaster, an Irish warrior familiar with the local terrain, and a Frankish smith to repair weapons. This constant mixing and mobility had profound genetic consequences. As these diverse crews traveled, they spread genetic lineages far and wide. A man of Baltic origin who joined a Danish crew might end his days settling in England, introducing his paternal line into the Anglo-Norse population. A Pictish woman captured in a raid on Scotland could be taken to Norway and become the ancestor of a new lineage there. The Viking Age was thus a period of intense genetic scrambling, breaking down ancient regional barriers and creating new hybrid populations. The longship was a vector not just for warriors and trade goods, but for DNA, carrying genes from the North Atlantic to the Caspian Sea and weaving them into a new, intricate European tapestry. The true genetic legacy of a Viking warband, therefore, is not one of purity, but of unprecedented mixture and connectivity, reflecting a world that was far more interconnected and dynamic than previously imagined. Beyond tracing ancestry and migration, the field of paleogenomics offers an unprecedentedly intimate window into the biological lives of the Viking Age people, revealing the diseases they suffered, the pathogens they carried, and the genetic adaptations that shaped their health. By sequencing the DNA of both humans and the microbes found within their remains, scientists can reconstruct a molecular history of health and disease that is invisible in the archaeological and written records. This approach moves beyond the grand narrative of kings and battles to the personal, often painful realities of daily life and death. One of the most significant discoveries has been the identification of ancient strains of deadly infectious diseases. Analysis of Viking Age skeletons from across Europe has found evidence of variola virus, the causative agent of smallpox. Sequencing these ancient viral genomes has allowed researchers to trace the evolution of one of humanity's greatest killers, suggesting that Vikings may have played a role in its spread throughout Europe during their travels. The presence of the virus in multiple individuals from different locations indicates that smallpox was likely an endemic and feared disease during this period, capable of causing devastating epidemics in non-immune populations. Similarly, paleogenomic analysis has uncovered the DNA of Mycobacterium leprae, the bacterium that causes leprosy, in Viking remains, particularly in Scandinavia and the Danelaw. By comparing the genetic makeup of these medieval strains to modern ones, scientists have found that the types of leprosy prevalent in Viking Age Scandinavia are similar to those found in medieval England, suggesting the disease may have been carried across the North Sea by settlers. The social stigma associated with leprosy was profound, and identifying its presence provides a poignant insight into the suffering of individuals who would have been cast out from their communities. The Vikings' own DNA also tells a story of health and adaptation. Studies have looked for genetic variants associated with immune system function, revealing how these populations were adapted to their local disease environments. Furthermore, analysis can identify genetic predispositions to a range of conditions, from metabolic disorders to hereditary diseases. While it is impossible to know if an individual with a specific genetic risk factor actually developed the associated disease, this information helps build a picture of the overall health profile of the population. For example, the prevalence of genes associated with lactase persistence, the ability to digest milk in adulthood, was variable across the Viking world, reflecting different degrees of reliance on dairy farming. Paleogenomics also sheds light on the transmission of zoonotic diseases, those passed from animals to humans. Given the close proximity in which Vikings lived with their livestock, often in the same longhouse, the potential for such transmission was high. Future research may identify ancient strains of pathogens like brucellosis or tuberculosis that originated in cattle and made the jump to their human owners. The vast trade and slave networks maintained by the Vikings also acted as conduits for disease on an intercontinental scale. A ship sailing from Dublin to Iceland could carry not only human passengers but a host of microbial ones as well, introducing pathogens to isolated communities with no prior immunity. The study of Viking health and disease is a stark reminder that their legacy is not just one of culture and ancestry, but also of biology and contagion. Their voyages connected the world in new ways, And this connectivity had consequences not only for the flow of goods and ideas but also for the spread of the microscopic organisms that have shaped human history as profoundly as any army. For centuries, our image of the Viking has been shaped by stereotypes. The towering, blonde-haired, blue-eyed barbarian, a dramatic and enduring but largely inaccurate caricature. The advent of paleogenomics has allowed us to move beyond these myths and reconstruct the physical appearance of Viking Age people with remarkable scientific precision. By analyzing specific segments of ancient DNA, ADNA, known to influence physical traits, researchers can now predict with a high degree of confidence an individual's eye color, hair color, and skin pigmentation, painting a detailed and surprisingly diverse portrait of the people of the North. This process, known as forensic DNA phenotyping, has been applied to hundreds of genomes from the Viking Age, yielding transformative results. One of the most striking findings from a major 2020 study was the revelation that brown hair was far more common than blonde hair among Viking Age Scandinavians. While blondism was certainly present and more common than in many other parts of Europe, the genetic data indicates a wide spectrum of hair colors, including various shades of brown, red, and black. This genetic diversity in appearance varied geographically. For instance, populations from the Danish regions showed a higher frequency of genetic variants associated with brown hair, reflecting their closer genetic ties to continental Europe. In contrast, individuals from the more northerly and westerly parts of Scandinavia, as well as the Swedish Vikings who traveled east, showed a slightly higher prevalence of lighter hair colors. This regional variation shatters the idea of a single Viking look, and instead points to a mosaic of different physical types across Scandinavia. The same diversity is true for eye color. While the genes for blue eyes were common, so too were the variants for green, hazel, and brown eyes. The classic combination of blonde hair and blue eyes was by no means the universal standard. This genetic reality aligns more closely with the nuanced descriptions found in some of the Icelandic sagas, which describe heroes and heroines with a variety of physical features, including dark hair. The analysis also confirms that Viking Age Scandinavians generally had fair skin, an adaptation to the low light conditions of northern latitudes which facilitates the production of vitamin D. However, the story of Viking appearance is not confined to Scandinavia. The constant movement and admixture of the era meant that the gene pool was continually being enriched with new traits. The influx of people from the British Isles, southern Europe, and the eastern Baltic into Scandinavia introduced a wider range of pigmentation genes. A Viking crew returning from a voyage to the Mediterranean might bring back not only treasure but also individuals whose DNA carried the genetic signature of a sunnier climate. Conversely, as Vikings settled in new lands, their pigmentation genes were introduced into local populations. The introduction of genes for red hair, which are found at high frequencies in Ireland and Scotland, into the Icelandic and Scandinavian gene pools is a clear example of this admixture. The ability to reconstruct the Viking visage from their DNA humanizes them in a powerful way. It allows us to look upon the reconstructed face of a 9th century Danish warrior or a 10th century Norwegian woman and see them not as mythical figures, but as real people in all their diversity. It replaces a monolithic stereotype with a gallery of individual portraits, revealing a population that was as varied in its appearance as it was in its ambitions, origins, and ultimate destinies. This genetic evidence forces us to see the Viking Age not in black and white, or simply blonde and blue, but in the full, complex spectrum of human variation. While the Viking genetic footprint across Europe is often a subtle layer added to an existing population, the islands of Shetland and Orkney, off the northern coast of Scotland, represent a different and more dramatic outcome. A near-total demographic transformation that made them a Norse genetic bastion for over a millennium. The settlement of these islands by Norwegian Vikings, beginning in the early 9th century, was not a mere elite takeover but a comprehensive folk migration that fundamentally and permanently reshaped the island's biological and cultural identity. Before the arrival of the Norse, the islands were inhabited by the Picts, a people known from enigmatic carved stones but who left no written records. The genetic fate of the Picts has been a long-standing mystery. While some undoubtedly intermarried with the newcomers, the genetic evidence from modern Arcadians and Shetlanders suggests a massive demographic shift. Unlike in Iceland or Ireland where the maternal lineages remained overwhelmingly local, Gaelic, in Orkney and Shetland, a very high proportion of both paternal and maternal DNA traces back to Scandinavia. Studies of the Y-Chromosome show that between 50% and 60% of male lineages in the islands are of Norwegian origin, one of the highest concentrations found anywhere outside of Scandinavia. Even more tellingly, mitochondrial DNA, mtDNA, studies reveal that a significant percentage of female lineages, perhaps as high as 40 to 50% in some analyses, are also Scandinavian. This balanced contribution from both sexes is the genetic signature of a large-scale family-based migration. Entire households, men, women, and children, must have made the journey from western Norway, seeking new farmland in the largely treeless but fertile islands. This process was so intensive that the pre-existing Pictish language and culture were completely erased, replaced by the Old Norse language, which evolved into the local Norn dialect, spoken until the 18th century, and Scandinavian legal and social structures. The islands became the seat of the powerful earldom of Orkney, a Norse polity that at its height controlled vast territories around northern Scotland and the Isles. This political and cultural dominance was built on a foundation of profound demographic change. The genetic distinctiveness of the Northern Isles has persisted for over 1,000 years. Modern inhabitants of Orkney and Shetland form a unique genetic cluster, clearly distinguishable from the populations of mainland Scotland, Ireland, and even from mainland Scandinavia. They are the direct descendants of this intense period of Norse colonization, a population shaped by centuries of relative isolation after the initial settlement. This isolation allowed the unique mix of Norse and residual Pictish genes to drift and evolve in its own direction, creating a distinct genetic identity. This legacy is not just a matter of scientific curiosity. It is a core part of the island's modern identity. The Norse heritage is proudly celebrated in festivals like Shetland's Uphelly AA, in the place names that cover the landscape, and in a strong cultural affinity with Scandinavia. When modern islanders take a commercial DNA test, the results often confirm this deep history, showing a strong genetic link to Norway that is far more pronounced than in any other part of the British Isles. Orkney and Shetland therefore stand as the prime example of what happened when Viking expansion met sparsely populated and strategically located land. It resulted not in a hybrid society, as in the Irish Sea, nor in an elite layer, as in Normandy, but in the creation of a new Scandinavian society in exile, a genetic and cultural echo of Western Norway that has endured with remarkable fidelity in the windswept North Atlantic. The Baltic Sea during the Viking Age was not a periphery but a vibrant and contested central arena, a liquid highway that connected the Scandinavian heartlands with the diverse peoples of the Finnic, Baltic, and Slavic worlds. This region was a dynamic frontier characterized by a complex interplay of raiding, trading, alliance, and settlement, creating an intricate genetic crossroads where DNA flowed in all directions. The genetic legacy here is not a simple story of Scandinavian expansion, but a complex tapestry of continuous interaction and admixture that profoundly shaped the populations on all sides of the sea. The island of Gotland, strategically located in the middle of the Baltic, exemplifies this reality. It was one of the wealthiest and most cosmopolitan centers of the Viking world, a semi-independent trading hub whose merchants amassed enormous fortunes, evidenced by the thousands of Arab silver dirhams found in buried hoards on the island. Ancient DNA from Gotland cemeteries reveals a startlingly diverse population. Alongside individuals of typical Scandinavian genetic profile are people with ancestry from Finland, the modern Baltic states, Latvia and Lithuania, Poland, and even further east. This genetic melting pot reflects Scotland's role as a nexus where different cultures met and mingled. A merchant from Gotland might take a wife from a Slavic trading partner in Volyn, in modern Poland, or form an alliance with a chieftain in Kurland, modern Latvia, leading to a constant influx of non-Scandinavian maternal and paternal lineages into the island's gene pool. The flow of genes was by no means unidirectional. Swedish Vikings, known as the Russ, famously traveled eastward, but there was also significant settlement and influence along the southern and eastern Baltic coasts. Archaeological sites like Grobina in Latvia and Wiskioten in the Kaliningrad Oblast show clear evidence of Scandinavian settlement, including characteristic boat burials and weaponry. These outposts served as bases for trade and the extraction of resources like amber and furs. Genetic studies in modern Baltic populations have identified a subtle but clear layer of Scandinavian paternal lineages, particularly haplogroup I1, attesting to the long-term presence of these settlers who eventually assimilated into the local Baltic and Slavic populations. The interaction also involved a significant and often brutal slave trade. The Vikings raided the Slavic and Baltic coastlines for captives, whom they called thralls. These individuals were sold in markets in places like Hedeby and Burka or taken back to work on Scandinavian farms. This forced migration represents a major vector for the introduction of Finnic, Baltic, and Slavic DNA, particularly maternal lineages, into Scandinavia itself. Ancient DNA from high-status burials in Sweden has sometimes revealed servants buried alongside their masters who were genetically of Eastern Baltic origin. This constant, often violent, interaction led to the creation of hybrid identities and frontier societies. The coastal peoples of the Baltic were not passive victims. They were active participants and formidable rivals. Groups like the Curonians and the Assylians, from the Estonian island of Saaremaa, were themselves skilled seafarers and raiders, who frequently attacked Scandinavian coastlines. This state of endemic warfare and alliance meant that genetic exchange was a constant feature of life on the Baltic frontier. The genetic legacy of the Viking Age in the Baltic is therefore one of complexity and reciprocity. It is a story of a sea that did not divide peoples but connected them, for better or for worse, leading to a millennium of shared history that is written into the DNA of the modern populations of Sweden, Finland, Poland, Russia, and the Baltic states. The genetic legacy of the Vikings extends beyond the demographic shifts in the general population. It is also deeply embedded within the very fabric of European aristocracy. Through a combination of conquest, strategic marriage, and dynastic ambition, Scandinavian DNA flowed into the ruling houses of numerous nations, creating an intricate web of kinship that connected the royal courts of Kiev, Paris, London, and beyond. This elite-level gene flow ensured that the Viking blood of chieftains and sea kings would persist for centuries at the highest echelons of European power, long after their descendants had abandoned the Norse language and customs. The most direct and well-documented example is the Rurikid dynasty. As established by genetic testing of their living descendants, the Y-chromosomal line of the founding princes of Kievan Rus traces back to a Scandinavian origin, likely from the east coast of Sweden. This single paternal lineage, which began with a semi-legendary chieftain Rurik in the 9th century, went on to rule vast territories in Eastern Europe for more than 700 years, producing the Grand Princes of Kiev, the Tsars of Muscovy, and a host of Russian noble families. Through this one dynasty, a specific Viking paternal line achieved extraordinary historical and genetic success, becoming a foundational element of Russian and Ukrainian statehood. In Western Europe, the path of Viking royal DNA began in Normandy. The Viking leader Rollo, granted lands in France in 911, established a dynasty that rapidly became one of the most powerful in Europe. His descendants, the Dukes of Normandy, intermarried with the French aristocracy, but they carried his Scandinavian Y-Chromosome. When his descendant, William the Conqueror, seized the English throne in 1066, he transplanted this Norse-descended paternal line onto English soil. The Norman and subsequent Plantagenet kings of England were, therefore, direct paternal line descendants of a Viking chieftain. Through the extensive intermarriages of their royal offspring, this lineage spread even further. Daughters of English kings married into the royal houses of Scotland, France, Castile, and the Holy Roman Empire, carrying their diluted Scandinavian autosomal DNA with them. The North Sea Empire of Knut the Great in the early 11th century provides another powerful example. As King of Denmark, Norway, and England, Knut presided over a vast, interconnected Scandinavian dynasty. His marriage to Emma of Normandy, who was herself of Viking descent and had previously been married to the English King Athelred the Unready, created a powerful royal family that blended Danish, Norwegian, and Norman Viking bloodlines. Although Knut's direct line of succession was short-lived, his relatives and the nobles he empowered continued to intermarry with other royal houses, further disseminating these genetic connections. The Viking presence in the Irish Sea also led to the creation of powerful Norse-Gaelic dynasties, such as the Y-Chromosome, who ruled Dublin, the Isle of Man, and parts of Scotland and northern England. These kings frequently formed marriage alliances with native Irish and Scottish royal families. The famous Scottish warlord Somerled, who drove the Norse from Argyll in the 12th century and founded the Lordship of the Isles, was himself of mixed Norse-Gaelic descent, and his descendants, Clan Donald and Clan MacDougall, became some of the most powerful families in Scottish history. Through these myriad connections, the genetic influence of a relatively small number of successful Viking leaders was magnified enormously, cascading down through the generations and across the political map of Europe, a lasting biological testament to their incredible political and military success. The explosion of the direct-to-consumer genetic testing industry in the 21st century has ignited a widespread and deeply personal quest for Viking heritage. Millions of people across the world, particularly in North America, the British Isles, and Australia, have sent saliva samples to companies like Ancestry DNA, 23andMe, and MyHeritage, hoping to uncover a tangible biological link to the romanticized and formidable world of the Vikings. This modern hunt for ancient roots has transformed the genetic legacy of the Vikings from a purely academic subject into a powerful component of contemporary personal and cultural identity, albeit one fraught with both exciting possibilities and significant misconceptions. The science behind these tests is twofold. Y-chromosome and mitochondrial DNA tests can identify specific haplogroups, broad genetic clans sharing a distant common ancestor. Discovering that one's paternal line belongs to haplogroup I1 or R1AZ284 provides strong, direct line evidence of Scandinavian ancestry, creating a powerful, almost tangible connection to the Viking Age. The second method, autosomal DNA testing, is more comprehensive but less direct. It analyzes hundreds of thousands of genetic markers across all chromosomes and compares an individual's DNA to reference panels of modern populations. The company's algorithms then estimate what percentage of one's ancestry comes from different regions, such as Norway or Sweden/Denmark. Receiving a result with a significant percentage from these regions is often interpreted by customers as direct proof of Viking descent. This has led to a cultural phenomenon where people proudly proclaim their Viking DNA, joining online communities, planning genetic tourism trips to Scandinavia, and sometimes adopting cultural symbols associated with the Viking Age. For many, this discovery provides a sense of connection to a deeper past, a narrative of strength, exploration, and resilience that they can incorporate into their own life story. However, this popular interpretation often oversimplifies the complex reality of genetics and history. A key challenge is distinguishing Viking Age Scandinavian ancestry from that of other historical Germanic groups, such as the Anglo-Saxons, Goths, or Franks, who share a deep ancestral heritage. The reference panels are based on modern populations, and while a match to modern Norwegians is suggestive of Viking ancestry, it is not definitive proof. The genetic signatures of different migrations from the same broader region can be difficult to disentangle. Furthermore, the percentage estimates can be misleading. A result of 10% Swedish does not mean a single Viking ancestor. It represents the cumulative, diluted inheritance from potentially hundreds of distant ancestors over 1,000 years. The commercial presentation of these results, often with dramatic maps and historical summaries, can encourage a simplistic reading of a deeply complex genetic inheritance. Despite these caveats, the modern hunt for Viking genes has had a positive impact by democratizing an interest in the past and in the science of genetics. It has prompted countless individuals to explore their family history, to learn about the Viking Age, and to appreciate the profound interconnectedness of human populations. It has also contributed to citizen science projects, where anonymized data from commercial tests can be used by academic researchers to map historical migrations with unprecedented resolution. This popular fascination represents the latest chapter in the long story of the Vikings' genetic legacy, a chapter where the echoes of ancient voyages are no longer just buried in the soil but are being rediscovered within the living DNA of their scattered descendants around the globe. On the northernmost frontier of the Viking world, in the vast subarctic landscapes of Fennoscandia, the Norse expansion encountered not fertile, settled farmland, but the domain of the Sami, a semi-nomadic indigenous people with a distinct culture, language, and genetic heritage. The long-term relationship between these two groups was complex, involving trade, competition, and cultural exchange, and it resulted in a significant and bidirectional flow of genes that is still visible today. This interaction represents a crucial, yet often overlooked aspect of the Viking genetic legacy, highlighting a story of coexistence and admixture on the Scandinavian peninsula itself. The genetic origins of the Sami are unique and ancient. They represent a blend of ancestral streams, including Mesolithic European hunter-gatherers and, crucially, a significant component of ancestry from ancient North Eurasians, a lineage that connects them to populations in Siberia. This gives them a genetic profile distinct from their Norse neighbors, who are part of the broader European genetic continuum. For centuries, the two groups lived in a state of symbiosis and tension. The Norse, who settled the coastal areas of Norway and the river valleys of Sweden, valued the Sami for their skills in hunting and trapping and for the valuable northern goods they could provide, such as fine furs, walrus ivory, and hides. The Icelandic sagas, like that of Egil Skallagrimsson, describe Norse chieftains traveling to Finnmark, the land of the Sami, to trade and collect tribute. This long-term contact inevitably led to intermarriage and genetic exchange. Analysis of both ancient and modern DNA has provided clear evidence of this admixture. Studies of the modern Scandinavian gene pool, particularly in northern Norway and Sweden, have identified characteristic Sami mitochondrial DNA, mtDNA haplogroups, such as U5b1b and Z, at low but significant frequencies. This indicates that Sami women were being incorporated into Norse farming communities over many generations. This process may have occurred through marriage alliances aimed at cementing trade relationships, or through the forced servitude of Sami individuals. The presence of these maternal lineages demonstrates a long history of social interaction that crossed the cultural boundary. The gene flow was not just one way. The Sami gene pool also shows clear evidence of Scandinavian admixture. Y-chromosomal analysis of modern Sami men has revealed the presence of typical Norse paternal haplogroups like I1 and R1a, indicating that Norse men also joined Sami communities. This may have happened for various reasons. A Norseman might marry into a Sami family, or an individual might choose to adopt the more mobile Sami lifestyle. This bidirectional exchange created a genetic cline, a gradual transition of gene frequencies, across northern Scandinavia, blurring the once sharp distinction between the two populations. Ancient DNA from Viking Age burials in Scandinavia has further confirmed this deep history of contact. A high-status Viking Age boat grave in northern Norway was found to contain an individual who was genetically Sami, buried according to Norse customs, suggesting that individuals of Sami origin could achieve high rank within Norse society. The Norse-Sami interaction is a vital part of the Viking story, demonstrating that the process of genetic mixing was not limited to overseas expansion but was also a fundamental feature of the Scandinavian homeland. It reveals a frontier that was not just geographical but also cultural, where two very different peoples forged a shared history that is permanently written in their respective genomes. While the DNA sequence itself provides a powerful record of ancestry and migration, a new frontier of genetic research, epigenetics, offers the tantalizing possibility of uncovering a deeper, more subtle legacy of the Viking Age. One written not in the genetic code itself, but in the molecular mechanisms that control it. Epigenetics involves modifications to DNA, such as DNA methylation and histone acetylation, that act as switches, turning genes on or off without altering the underlying sequence. These changes are profoundly influenced by an individual's environment, diet, and lifestyle. The central and most speculative question is whether some of these environmentally induced epigenetic marks could be passed down through generations, meaning that the biological experiences of Viking Age ancestors might still be subtly influencing the health of their descendants today. The Viking lifestyle was one of extremes, creating strong environmental pressures that would have undoubtedly shaped their epigenomes. Their diet, for example, varied dramatically by region. Coastal Norwegian populations consumed vast quantities of cold-water fatty fish, rich in omega-3 fatty acids, which are known to influence epigenetic markers related to inflammation and cardiovascular health. Inland agricultural communities, by contrast, had a diet based more on grains and dairy. It is plausible that these distinct diets left different epigenetic signatures on genes related to metabolism. Similarly, the physical demands of life, rowing a longship across the North Atlantic, farming in a harsh northern climate, or engaging in frequent combat, would have induced epigenetic changes related to muscle development, stress response, and energy utilization. Periods of famine, a recurrent threat, are known to cause significant epigenetic shifts that can, in some documented cases, have health consequences for subsequent generations. For instance, studies on the descendants of individuals who survived the Dutch hunger winter of 1944–45 have shown that prenatal exposure to famine resulted in epigenetic changes that were still detectable decades later, and were associated with a higher risk of conditions like obesity and diabetes. While directly proving a similar transgenerational epigenetic inheritance from the Viking Age is currently beyond the reach of science, it remains a compelling theoretical framework. Could the descendants of Norse Greenlanders, who endured centuries of a marginal and deteriorating climate, carry epigenetic marks related to metabolic efficiency? Could the populations of the North Atlantic, descended from people who relied heavily on a marine diet, have inherited epigenetic settings that affect how their bodies process fats? The challenge lies in distinguishing any such ancient legacy from the accumulated epigenetic changes of the intervening millennium. The epigenome is dynamic and constantly responding to new environmental inputs. However, as our ability to read and interpret epigenetic information from ancient remains improves, it may one day be possible to reconstruct the epigenetic landscape of a Viking Age individual. By comparing these ancient epigenomes to those of modern populations with known ancestry, researchers could begin to identify long-lasting epigenetic patterns. This field remains highly speculative, but it opens up a profound new dimension to the concept of genetic legacy. It suggests that our inheritance from the Vikings may not just be the DNA sequence they passed down, but also a faint molecular memory of their struggles, their diet, and the very world they inhabited, a biological echo of their lives that continues to resonate within our cells. The study of the Viking genetic legacy, already a field transformed by revolutionary discoveries, is poised on the brink of another, even more profound era of exploration. As technology advances and methodologies become more sophisticated, the future of Viking genetic research promises to move beyond broad population histories to incredibly detailed, individual-level narratives, reconstructing the Viking world with a resolution that was once the exclusive domain of science fiction. The primary driver of this future will be the sheer scale of data. The sequencing of a few hundred Viking Age genomes was a landmark achievement. The next step will be to sequence thousands, and eventually tens of thousands. This massive increase in sample size will allow for fine-scale mapping of genetic variation across time and space, revealing subtle, local patterns of migration, kinship, and social structure. It will become possible to reconstruct the detailed genetic history of a single parish in the Danelaw or a specific valley in western Norway, identifying the precise origins of its settlers and tracing their family lines through centuries. The integration of ancient DNA with other scientific techniques will create a more holistic understanding of Viking lives. Combining genomic data with isotopic analysis of teeth and bones, which reveals an individual's diet and geographic movements throughout their life, will allow researchers to create detailed biographies of people who left no written record. We will be able to identify a first-generation immigrant in a Viking town, determine their exact genetic ancestry, know what they ate as a child, and trace their journey from their birthplace to their final resting place. This fusion of data will bring the past to life with unprecedented clarity. New analytical techniques will also unlock more information from the DNA itself. The study of the Y-chromosome and mitochondrial DNA will be refined to trace lineages with pinpoint accuracy, while analysis of the X chromosome will offer new insights into female-specific migration patterns. The burgeoning field of paleoproteomics, the analysis of ancient proteins, will complement DNA evidence, providing information about health and disease that may not be preserved in the genetic code. Furthermore, the recovery of environmental DNA, EDNA, from archaeological sites holds immense potential. By extracting and sequencing DNA fragments from soil layers in a Viking longhouse or the sediments of a harbor, scientists can reconstruct the entire ecosystem of the site. They can identify the plant species that were being farmed or foraged, the types of livestock being kept, and even their genetic origins, the pests and pathogens present in the settlement, and the composition of the human population, all without needing to find skeletal remains. This will provide a complete biological snapshot of a Viking Age community. Ultimately, the future of this research lies in its power to tell more human stories. It will move beyond simply asking where did the Vikings come from, to answering more personal questions. Who was this specific woman buried in Burka? What was her genetic ancestry? Did she have children, and can we find their graves? What diseases did she carry? By weaving together multiple strands of scientific evidence, the future of Viking genetic research will continue to strip away myths and replace them with the complex, diverse, and deeply personal realities of the people who defined one of history's most dynamic eras. This narrative traced the Vikings' genetic trail across Europe, from their Scandinavian origins to their settlements in England, Ireland, and beyond. It revealed how their DNA tells a story of migration, admixture, and the creation of new identities. Please let me know what you think and how it could be improved.